Namaskaram. Today in this session, uh, the topic is anomalous secondary growth in monocotyledonous stem. So, generally in angiosperms, it is classified into two major groups. One is monocotyledons and another one is dicotyledons. And generally, the dicotyledonous plants show the normal secondary thickening, and also it shows the anomalous secondary thickening also. In certain species, we can see the abnormal activity of cambium, which develop irregular nature of production of vascular tissues, and which leads to the changes or ornamentation become varied from the normal secondary thickening which is taking place. So the morpho-anatomical features in a normal dicot stem showing secondary thickening is changed or altered, as in some of the members coming under dichotomous plants, and which are commonly called as the anomalous or abnormal behavior of secondary thickening due to the abnormal behavior of vascular cambium. So here in this topic, we have to understand that even monocotyledonous plants, generally these members do not show a wider range of secondary thickening or anomalous secondary thickening. But some of the members which show the secondary thickening in monocotyledons, and similarly coming to the stem, which show that is in monocotyledonous stem, which is some of the species or members which show the anomalous secondary growth in the whole group. Okay, so here in the present session, we are taking one of the example as Justina. Justina is one of the monocotyledonous dicot monocot stem or the plant which show the abnormal activity of secondary thickening. Okay, so as in the young condition in Dracena, even it shows the normal nature of development of vascular bundles, and they are collateral and closed bundles or open bundles which is seen in monocots. And here in Dracena, we can see that a closed primary vascular bundle showing both the xylem and phloem, and a number or numerous number of such closed collateral bundles which is getting disposed inside the ground tissue in the young condition. So it is the normal development of the stem, and after a while the plant which show the secondary growth in thickness, the early formed or primary formed tissue, particularly the vascular tissue, which consists of both the xylem and phloem, and devoid of fascicular cambium, and so that is why the bundles are closed and which is disposed in the ground tissue. And coming to the anomalous secondary growth, what happened? The secondary growth is rare in monocots, and the derivatives of primary thickening meristem (ETM) from ground parenchyma traversed by procambial strands. So the early formed young bundles or early formed vascular bundles which are closed and collateral, which is dispersed in the ground tissue. And outer to these early formed or primary bundles, they are the ground tissue, which show the parenchymatous cells, which is dispersed in the outer region, and the cortical region, which show the formation of such cambial cells. And coming to that, previously we can understand about that the secondary growth is where monocot is the first one, and second one derivatives of primary thickening meristem. Derivatives of primary thickening meristem. This meristem which is developed in the outer cortical region, or otherwise there is no such a differentiation in monocot, cortex or vascular zone, because the early formed or primary bundles which are getting scatteredly dispersed in the ground tissue. So outer to, somewhat outer to the early formed vascular bundles, there the primary thickening meristem formed in the ground parenchyma. And such primary thickening meristem or PGM, which act as the procambial strand and which differentiate into vascular bundles. So such primary thickening meristem is getting differentiated in the ground tissue outer to the early or primary formed vascular bundle, and which become meristematic in nature and divide and produce certain vascular tissues. And so such procambial strands are developed outer region, that is in the ground parenchyma, and which later differentiate into the vascular bundles. And actually, or normally, the vascular bundle consists of possessile and phloem. And we are coming to the secondary growth. The secondary growth initiated in the inner cortex, and the formation of multi-layered cambium is the first the related process in secondary growth and thickness in monogamous plants. So the formation of multi-layered cambium, and these are cambial strands which is formed in the ground parenchyma, which are show irregular nature, and they are irregular in size, and intermittently those for multi-layered cambium which divide to produce such vascular bundles, that is the second and the third row of vascular bundles. And usually such bundles which are formed from the primary thickening meristem, that is, it is from the multilayered cambium which is developed in the ground tissue as the part of anomalous secondary growth in thickness. Here we can see such bundles are placed in the outer regions of justicia, that is the stem of justicia. They are alternating bands of concentric rings of bundles. That is, it is clearly formed as a concentric level in the outer cortex or outer to the early formed vascular bundles. And such alternating bands of concentric rings of bundles which is formed. Why these alternating bands? Because the second row of bundles and third row of bundles or subsequent row of bundles which are formed in the ground parenchyma from the multilayered cambium, they are oriented in such a fashion, that is alternating bands, because the alternating area or locations of the multilayered cambium which develops or derive the vascular bundles, that is phylum and phloem. And coming to the nature of abnormal behavior of vascular cambium is seen, it is of due to such alternating bands of concentric bundles which are formed in the outer region of ground tissue in justicia. This is one of the plants. And coming to the nature of, in connection with the formation of secondary growth and thickness in monocot stem, example justicia. Here we can see in addition to the ground region, the peripheral region of the stem or the outer zone of the stem, which shows the formation of or cambium. So here it is in monocots there are no stellar region or cortical region or the outer region such a clear differentiation as seen in dichotomous plants. And here in monocots or in monocotyl plants only we can see the ground tissue, the entire internal area, and there is a delimitation of outer zone. And that outer zone in the primary structure it is derived as epidermal tissue system, and it contains the hypodermal collenchyma and some bands of scleronematous cells. But due to the secondary growth which starts what happened normally or in connection with the secondary growth or anomalous secondary growth in thickness starts in monocots, the outer hypodermal collenchyma or epidermis or those bands of sclerenchyma which is getting demolished. And so, in connection with the secondary growth or anomalous secondary growth in monocot, here in Dracena we can see the formation of orthocymbium. So, there are three stages of anomalous secondary behavior or growth in Dracena or in this monocotyledonous plants. We can remember that. First one, the normal bundles which are formed in the primary structure which is dispersed or distributed in the ground tissue, and outer to these primary bundles those second or third row of vascular bundles which are derived from the primary thickening meristem from the ground parenchyma. So it is the first process, and second process this PGM primary thickening meristem is developed in the ground parenchyma, which develops or differentiated to form the multi-layered cambium. And those multi-layered cambium are irregularly in shape and size, and which produce the second process in the anomalous secondary growth, that is from that multi-layered cambium it produces the alternating bands of concentric rings of vascular bundles as second or third row. Okay, and the third one is in connection with the secondary growth in this species or in this plant or in this monocotyledonous plant with the formation of fourth cambium, and ultimately it is also the part of secondary thickening in monocot stem. And here the formation of fourth cambium, which as similar as in dichotomous plants, the fourth cambium which produce outer phloem and inner phloem and middle phloem and inner phloem. So phloem, 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 that is the core of the cambium and the secondary cortex which is derived from the formation of fourth cambium in connection with the secondary growth and thickness in monocots. And this is the nature of the anomalous secondary thickening which is seen in monocotyledonous plants of the kingdom. Okay, and here in this Dracena, another peculiar nature of the secondary or tertiary ring of alternating bands of vascular bundles which are formed and are amphivascular vascular bundle. That is, those bundles which is formed during secondary growth and thickness, it contains or the structure of the vascular bundle is in the center it is completely filled with the phloem, and the phloem is completely surrounded by the xylem elements. Such bundles are called amphivascular vascular bundles. So this is the general nature of anomalous secondary growth and thickness in monocotyledonous plants. Example, Dracena. Thank you.